Good afternoon, everyone. Thank you so much for being here today. We so appreciate you being with us today in the midst of the COVID-19 pandemic and a tough year for everyone. We wish we could all be together in person and look forward to when we can. A quick reminder that the chat function is disabled and that all of you are on mute without video, but please use the Q&A to submit your questions. There are two buttons at the bottom of your screen. Please use the button marked Q&A to submit live questions to our speakers. If we don't get to your question, we will follow up with you after the event. I'm Meg Cruz from Lucille Packard Foundation, the sole organization responsible for fundraising for Packard Children's Hospital and Stanford School of Medicine. We partner with Stanford Diabetes Clinic and the research program to raise funds for this important mission. So let's get started with the program. Today we're going to hear from Dr. David Maas, who will highlight the incredible growth of the diabetes field at Stanford, which led to our national recognition as a top 10 program by US News and World Reports. Then we will hear the story of why Dr. Anna Gloyn moved here all the way from Oxford, and she'll share key insights about her work unraveling the mysteries of diabetes and our genes. Our first speaker today will touch on innovations in the program, such as hiring diabetes educators for the new onset diabetes program. This amazing program helps newly diagnosed patients and their family members with education and training to safely manage diabetes. One of the most well-known faces in the clinic, Melissa Anderson, is a special volunteer and a mother of a daughter with diabetes, here on the screen. She is the lead parent mentor in the family-centered care department at Packard Children's Hospital. Welcome, Melissa. Thank you so much for being here today, and please take it away.
Thank you, Meg. I'm a parent of an adult daughter with type 1 diabetes. I'm a proud member of the diabetes community, and I'm honored to be here today to introduce Dr. David Maas. My extended family's experience at Stanford has a long history that dates back to 1994. I have both a niece and a nephew who received life-saving care here at Stanford before diabetes became my immediate family's reality. The care our daughter has received at Stanford, and including the diabetes team's willingness to approach that care from a patient-centered perspective. Our daughter was among the first in the world to use an early closed-loop system, and she has participated in many studies conducted at Stanford that aim to bring a more effective and patient-centered approach to blood sugar management. I started my role as a parent advisor within the Family Centered Care Department at Stanford Children's in 2013. I already knew of David because of the Pink Panther book, which many of us received in clinic when our kids were diagnosed. David's a co-author, but I didn't know about David's leadership in ISPAD, the International Society for Pediatric and Adolescent Diabetes, or his keen understanding of the burden diabetes places on families and children. When David first arrived at Stanford, we met for coffee, and I'll never forget his passion as a patient-centered doctor first and a scientist second. He cares deeply about his patients and their families and wants to see them flourish despite living with diabetes. Within the endocrine department, David has developed a team of diabetes professionals who holistically approach care for patients. He began the digital health program in 2017, well before COVID made telehealth a necessity, and he has championed numerous efforts to improve access to care for the most vulnerable of patients. He is a mentor and leader in the diabetes community, and I'm honored to introduce Dr. David Maas, Division Chief of Diabetes and Endocrinology at Stanford Medicine. Take it away, David.
Thank you, Melissa. Thank you so much for the kind words. Also want to thank and welcome everyone who's attending, zooming in with us today. Want to thank Meg and the foundation for all of the work that you've been doing, putting this event together. And then also want to especially thank Dr. Anna Gloyn, who we're going to hear from shortly. But first, I'm only going to speak for about 15 minutes so we can get on to Anna's presentation. But I would like to share with you some of the things that I'm most excited about, some of the advances we've made in the diabetes program at Stanford in the last few years. But before that, I even want to go back and talk a little about some history with diabetes, and then also a history of how I got involved with diabetes from a personal standpoint. So people have probably seen this picture before. This is a picture of Banting and Best, and we're coming up in 2021 on the 100th anniversary of the discovery of insulin. At the time, this was a remarkable discovery. It was considered a miracle cure that you could have now a treatment for what had been a deadly disease. And as we look at this picture, we see young Teddy Ryder, who is on the left, he was very sickly, thin looking before he was able to get treated with insulin. And then you can see a few months later that he then is quite pleased with himself and doing very well on his tricycle in 1922. So even better, however, is that we have a picture here of Mr. Ryder in his 70s, that he lived a long and happy life, being able to care for himself with insulin. But we know from the stories, and we know from our even modern day experience, that insulin's not a cure for diabetes, it's a treatment. And so it was able to transform what was a deadly disease into a chronic disease. But it also, as we see with this next picture, became a great burden on people with diabetes and on their families. This is a picture from a book called Bittersweet. It's a history of the Joslin Diabetes Center. And this is a picture, one of the patients in the 40s, 50s, and 60s. He was an artist, and he would bring pictures into his doctor. This is what diabetes care was like to him. You can see what looks like a very elaborate chemistry experiment, which was how you had to try to measure roughly what your glucose levels were. And then I think most striking is you can see what in his mind it was like to give an insulin injection at the time. When I saw this picture, it was brought home memories to me of my grandfather. And here's a picture of me as a child with my sister Sarah, my Aunt Katie, and then my grandfather. And I remember as a child watching him sharpen needles. And I remember how much he disliked having to give his insulin injections. So we've come a long way from those days, and we'll talk a little bit about that. But this sparked my interest in diabetes. And when I went to medical school, then my first opportunity to have a clinical experience, I chose to go to the diabetes clinic at the Indian Health Center here in Albuquerque. And that furthered my interest in diabetes and learning more about how to care for people and the burden that people carry with having to care for their diabetes. From there, after completing my pediatric residency, I went on to do 3 years of fellowship training in pediatric diabetes at the Barbara Davis Center. Had wonderful training there in both research and clinical care. As Melissa mentioned, I had the opportunity and honor to help co-author Understanding Diabetes, or what we know as the Pink Panther book, with Dr. Peter Chase. It was also at this time that a lot of the research in artificial pancreas was beginning. And with that, I had the opportunity I was at the University of Colorado to work with Drs. Wilson and Dr. Buckingham at Stanford. And so it was about 5.5 years ago when Dr. Wilson approached me and said he was going to be stepping down as the division chief, and if I might be interested in coming to Stanford. And of course, was honored that he would think of me that way, and also very intrigued with all of the potential that there is at Stanford, with what a strong program existed already, and some of the friendships and relationships I already had. So my goal in coming to Stanford was to build upon this very strong program. What can we do more? How can we deliver better care? How can we expand the research that we're doing? how can we grow the team that we have to do more and more. So with this next slide, it looks very busy, but basically what I'm trying to show here is that over the past 100 years, there have been advances in glucose monitoring, first urine testing, then blood glucose, and now we're all well aware of the era of continuous glucose monitoring. At the same time, insulin delivery has progressed from those insulin injections that we saw in that sketch to the first insulin pumps, which have become better and better and smaller and smaller. And now we're in the area of the continuous glucose monitors talking to the pumps, which are now smart enough with algorithms in them to help dose the insulin. So we're very focused on getting tighter glucose control. We're very focused on reducing the burden of disease. And these are a couple of the things that the artificial pancreas is already bringing to our patients after this decades of research. So we're quite pleased at Stanford that we're the only center involved in all 4 of the big NIH-funded artificial pancreas studies, and we have Drs. Buckingham and Dr. Hood to thank for their leadership in this area. Dr. Buckingham's the site PI for 3 of these studies, and then Dr. Hood is leading the psychology work on all 4 of those to try to figure out how can we make these systems work better for children, young adults, and adults with diabetes. So, as I said, we're very focused on getting better glucose control, reducing the burden of care that's associated with diabetes, but we also have to remember what kids want, and kids want to have fun. So this is a slide from an artificial pancreas ski study that we did. We combined a research study with kids being able to go ski. Here's a picture from when we did it in Colorado with colleagues from the University of Virginia. We repeated that study up at Kirkwood a couple of years ago, also with colleagues at both of those institutions. And the kids really had a fun time. Also, we showed that these systems can work well at altitude, at cold, with vigorous activity, and the kids really had fun. So this was a fun study trying to push the limits, trying to do cutting-edge research and show that these systems worked. But in addition to doing the cutting-edge research that we're well known for at Stanford, we're also very keen on moving all of these technologies into the clinic so that it's available to everyone. And so this leads to a program we recently initiated called the 4T program. And what this slide is, the graph shows what historic hemoglobin A1Cs have been in a clinic. At diagnosis, it's often high. We know that because people haven't been diagnosed yet. And then as treatment begins and children go into the honeymoon phase of disease, their A1C comes down. However, then it starts drifting back up as the challenges of diabetes and the natural history of losing the beta cell function cause it to be more difficult to have tight glucose control. So what we'd like to do is to flatten this curve out and keep that A1C down at 7% or lower, because we know the long-term benefits of that for health. So in this program, we call it 4Ts or teamwork, and that's also teamwork between the clinic and the families, but also within the clinic, the doctors, the diabetes educators, the psychologists, the dietitians, that we're all working together for your child's health. Technology, we're starting technologies, continuous glucose monitors very early. We offer those in the first week and try to get everyone started in the first month. And then to have very clear targets so that people know what to do. So that first part of the study is to get the technology early. The second part of this study, we're working with our psychologists because we want to optimize quality of life, and Dr. Corey Hood's leading that part. And the third part of this study, I picture David Schenker, who is a PhD in mathematics in the engineering school, and he's working with us on algorithms so that we can take all of this data that we receive from the CGMs and try to use it in a more precise way to communicate with families, to be able to let them know when dose adjustments are needed, and to do it in a more timely way than every 3 months with scheduled visits. Also working to try to scale this program up so that we can reach more people and help more people with this program. It's not all glucose control, we know that. we're very pleased, and I keep talking about the things I'm most excited about. Something I'm very excited about is that we have 4 diabetes psychologists, Corey Hood and Deanna Ranjo, both professors, and then we've got Molly Tannenbaum and Jesse Wong, who are both more recent additions to our program, and they're available not only for research, but also to help children and their families with some of the strategies to help deal with the distress that can come along with diabetes. So Melissa mentioned a little bit about the digital health and how we're able to move quickly when COVID hit us to be able to transition to more telehealth. And so that's something that we will likely continue, most certainly continue to do in the future. We're able to get the downloads from devices so that we're able to have that data and then carry on visits via telehealth. So one of the things that Melissa also mentioned was my role with ISPAD, or the International Society of Pediatric and Adolescent Diabetes. And from that, we learned a lot from our international colleagues, many of whom are doing a better job than we are in the US with getting better glucose control for their children. And so one of the things that we've done with that is joined an international diabetes registry. It's called the SWEET Registry, and we're very pleased that we were the first North American pediatric diabetes center to gain the recognition of a center of reference by the SWEET International group. And also in this past year that we won the Best Improvement Prize, and Dr. Priya Prahalad has been leading our efforts in that. So this international recognition has been very gratifying. Locally, another thing I'm very excited about is with the leadership of Dr. Sung Kim, who is in developmental biology and does basic science research on islets and other diabetes-related research, that in 2017, we were one of 16 centers to receive an NIH research grant as a diabetes research center. And the reason for these are to accelerate basic and clinical research in diabetes. Over the past few years, we've grown from 100 members to 120 members in the Stanford community who are members and doing research. Some are new people who are recent graduates. Others are established investigators who, because of this center, have become more interested in doing diabetes research. We've got involvement of 3 schools at Stanford, medicine, engineering, as well as humanities and science. And we also have the support of both the adult and the pediatric hospital. So this is truly an across-campus effort, and we're making great progress with this. So I think the next slide is perhaps what I'm most excited about, that we are training the next generation of clinician-scientists. We have 3 wonderful mentors, Dr. Hood, Dr. Buckingham, and Dr. Gloyn, but we have 6 young trainees, physician or psychology scientists who have completed their training and now are on these career development awards. It's very prestigious. NIH does not give out many of these. It's rare for a program to have more than 1 or 2 people on these awards, and we're very pleased that we've got 6 very bright, hardworking young scientists on these awards, as well as having wonderful mentors to help guide them on their way forward. So I started with a picture of 100 years ago, Banting and Best and Marjorie the dog, and the discovery of insulin. In the past 100 years, we've learned so much more. We know more about islet cells, beta cells. We know more about stem cells, immunology, bioengineering, and genetics. And all of this increased knowledge is going to help us improve care for people with diabetes, reduce the burden that people have to suffer from with diabetes. But also, we want to work towards preventing diabetes. And ultimately, we want to cure diabetes. And so with that, I'm going to now introduce Dr. Anna Gloyn, Professor Anna Gloyn, who joined us from Oxford. Anna is a world-renowned researcher in diabetes genetics. She's a wonderful mentor. She is an outstanding teacher and has a great gift of being able to take very complicated science and explain it clearly. And Anna, I'm so delighted you're here. Anna's a wonderful colleague. It's really been lovely to get to know you, and just greatly appreciate all the work you've done so far and all the great work that lies ahead. So thank you very much, Anna, and thank you all for your attention.
Wonderful. Thank you so much, David, for that very warm introduction. I am absolutely delighted to be here this afternoon and to have the opportunity to share with you a little bit about my diabetes research journey, which has recently brought me to Stanford. I hope you enjoy hearing about some of the work that I've done in the past, and also hearing about some of the work we're currently doing here at Stanford and my vision for how we can build on the fantastic work David and his team are embarking on here at the Stanford Diabetes Research Center. So my research journey really started back in 1996, and I'd like to take you through how I got in 2020 to arriving here in sunny California. So why do I work on diabetes? Well, as a scientist, I guess I could have picked any biomedical trait to have invested my time and effort in. But what really drew me to working on diabetes was, as an undergraduate back in 1992 at the University of Surrey, in the first week I was on campus, I met up with 3 other girls who shared my passions for science and for socializing, and the 4 of us made a long-lasting friendship. One of these very good friends, Helen, happens to have type 1 diabetes. And this was really the first time I had come into close contact with anyone who was experiencing life with diabetes. And I have to say, it was an enormous wake-up call, not just for myself, but for Jenny and Sarah as well, as we tried to help Helen throughout her university career, managing her diabetes. We got to learn about her family, her brother and her mother, who also have type 1 diabetes, and we're able to discuss how Helen felt about living with diabetes and the implications it had for her and her career, and also her dream of being a mother. But in addition to learning about the very personal side of diabetes from Helen, I also was able to learn about the biochemistry of insulin in the classroom. And I guess it was recognizing the impact of the pancreatic beta cell and its in diabetes, and what the hormone insulin does in our body, that sparked an interest in dedicating my research career to understanding the pancreatic beta cell and what goes wrong in all types of diabetes. So this prompted me to search for opportunities to train with world-leading experts in pancreatic beta cell dysfunction in diabetes. And I was very fortunate to be taken on by Professor Robert Turner at the University of Oxford. Robert was a pioneer in the field who led one of the largest clinical trials, understanding how important glycemic control is for controlling your risk of diabetic complications. And I spent 4 wonderful years in Oxford learning about the molecular genetics of type 2 diabetes. So for the last 20 years now, my work has really focused on capitalizing on discoveries that have arisen in nature. So these are genetic variants that we know are causal for monogenic forms of pancreatic beta cell dysfunction that cause diabetes, or more recently, common genetic variation that's present in the general population that increases your risk of developing other forms of diabetes, like type 1 or type 2 diabetes. And what my research involves is using these spelling mistakes in our DNA, excuse me, as tools to interrogate the mechanisms that stop our pancreatic beta cells excuse me, functioning properly and secreting the hormone insulin. Apologies for that. I can assure you it's just a cough and it's not COVID. I'm recovering from a long cough that's been here with me for 5 weeks, but I'm going to soldier on. So, yes, as I say, my work centers around using these variants as tools to interrogate cellular and molecular mechanisms of type 2 diabetes risk in particular, and monogenic forms of pancreatic beta cell dysfunction. And what I like to be able to do is to translate what I learn about how a pancreatic beta cell works, how that beta cell makes and secretes insulin, what keeps that beta cell alive and functioning, and to translate that into the clinic so that my colleagues like David can use that information to improve how they treat patients with all varieties of diabetes. So in February this year, I made the decision to hop on a plane and leave Oxford and join David and his team here at Stanford. And that was a big decision for me and my family, but it was a very easy one to make because Stanford was such an attractive center to start to build the rest of my career in. It has an award-winning center for genomics and personalized medicine, and also this phenomenal diabetes research center that brings together basic scientists across many different disciplines and puts us adjacent to clinical colleagues who are tackling diabetes on a daily basis in the hospital setting. Words that come to mind when I think about Stanford are translational research, innovation, state of the art, and all of these things really attracted me to being part of this environment where I would have the opportunity to take things that I developed in the laboratory and to see them impacting on patients' lives within my lifetime. So why do I work on genetics? Why do I think this is a really powerful way of understanding why pancreatic beta cells fail in all types of diabetes? Well, an example that I like to give is work that I was involved in as a postdoctoral researcher in the United Kingdom. And during my postdoc, I identified mutations in a really important component of the machinery that couples your sugar metabolism to the ability of your beta cell to release insulin after a meal. And children like Lily, who have this form of monogenic diabetes, are unable to secrete insulin in response to a meal. So Lily had been treated by insulin injections, but on the basis of the molecular diagnosis that we were able to give her, and building on the work of others who had already identified sulfonylureas as a type of drug that can close this channel, we were able to transfer Lily from an insulin injection onto an oral tablet to treat her diabetes. And not only did this have a huge impact on her quality of life, as I'm sure you can appreciate, it also had a really positive effect on her glycemic control, and her blood sugars were the best they had ever been in terms of their control. So this is an example of precision medicine, giving a person a particular type of medication on account of their individual characteristics. But it's also an example of how human genetics can identify a safe and effective target for therapeutic development for a broader population of patients, because sulfonylureas are actually used to treat patients with type 2 diabetes. So how is my team using human genetics at the moment? Well, in recent years, we've seen astonishing progress in our ability to understand the human genome. And at the moment, there are over 300 regions of our genome that we know influence your risk of developing diabetes. And what my group is doing is using these clues in our genome to help us identify safe and effective drug targets, to improve islet cell replacement strategies, and to extend our ability to offer precision medicine to a wider population of patients. And what I want to do now is give you a sense of what I mean in each of those three areas and to relate it to work that's actually going on at the moment in my lab. So let's start by thinking about how we might use human genetics to identify safe and effective drug targets for pharmaceutical companies to make new molecules against. Well, a really nice example of how we can use human genetics in this space comes from work that I published with a number of collaborators last year. And this was where we used a discovery that we'd identified rare variants in the human genome that protected you from developing type 2 diabetes. And these variants were in a zinc transporter in the pancreatic islet. And by understanding that these variants protected you against diabetes, we were able to uncover a piece of machinery in the beta cell that nobody really thought of as a potential drug target before. And why we were really excited about this, and not just us, pharmaceutical companies were also excited, was because it takes pharmaceutical companies a really long time to get a drug from the laboratory into patients. And it's a very expensive process. And most of the molecules that they start in clinical trials that have worked in a laboratory setting in animals and in a dish, they don't work when you put them into patients. And the reason for that is because they are toxic with some undesirable side effects, or they don't work. So by looking at the human genetic data, the pharmaceutical companies realized that they could get a heads up on whether something was likely to be effective in a human, and they could also identify whether it was likely to have any adverse side effects. And with our zinc transporter, they could see that it was going to be effective in humans, that it was unlikely to have any side effects. And through the work that we did in my laboratory, they were able to determine that they should make an antagonist rather than an agonist. And now a number of pharmaceutical companies have programs directly working on this new drug target, which we hope will bring a new treatment to the clinic in a number of years. So the second way that we can use human genetics to improve our understanding of the beta cell is that I'm sure many of you are aware that there's an enormous amount of excitement about the possibility of regenerative medicine, making beta cells in a dish and being able to transplant those into patients with type 1 diabetes. And what we want to be able to do is to help our colleagues with those protocols so that they can make the best possible beta cells for this stem cell replacement strategy. And the reason why this is exciting is, I don't know if you know, but most of what we know about how we make a pancreatic beta cell comes from studying mice. And it turns out that mice and humans are actually quite different. And there are some really important differences in how a mouse makes its beta cell and how a human makes its beta cell. And human genetics is one way that we can learn about those differences. So what I'm doing at the moment is trying to use human genetics to tell us about how we can make a beta dish most effectively, help people making protocols for stem cell replacement strategies to make the best beta cell possible. I thought it might be quite interesting to tell you a little bit about what that actually means in my lab. And I'm going to give you an example here of an experiment that's actually happening at the moment in our lab. And that's where we've taken a gene that we know is really important for diabetes risk. And we've used a technology that I'm sure you've heard about in the lay press at the moment called CRISPR, which of course has been the Nobel winning prize this year. And we've used CRISPR to cut out this diabetes gene from a human iPS stem cell. And then we've taken this stem cell in a dish and forced it to become a beta cell. And what we've learned from this experiment is that if you take the REB1 diabetes gene out of the stem cells, they actually make better beta cells. And this is really exciting to us because it's telling us that there are pathways that we can manipulate within the cells that will help them to make more efficient pancreatic beta cells that could improve current protocols. Okay, so what about that final area of research? I'm going to turn my attention now to how we can use human genetics to enable precision medicine. So that's something that you'll often hear about in the press, and it's really about giving a patient the best drug treatment at the best time that's best suited for that particular individual. And let me tell you how I think genetics can help us in that regard. So for many years now, we've been actually doing precision medicine for diabetes, but mainly for single gene disorders, these rarer forms of diabetes that are due to a spelling mistake in a single gene that result in diabetes that perhaps causes defects in the way your beta cells sense glucose, how your beta cells are made, the KTP channel defect that I told you about with Lily, and also mutations that alter how you put fat down in your body. And by understanding those different varieties of diabetes, we've learned an enormous amount. And we can also now give patients with these particular varieties of diabetes tailored treatments because we understand that they work best for these different disease mechanisms. But what's really exciting at the moment, because of the enormous amount of genetic data that we have now, is we're starting to realize we can use common genetic variation in a way we didn't originally anticipate to give us clues about how we might be able to deploy existing therapeutics most effectively for people with all types of diabetes. So I'm going to share with you some work that we published last year, and also some work that we haven't published yet that we're really excited about. So a couple of years ago, we identified some DNA changes in a gene called PAM that were associated with increasing your risk of diabetes. Now, as it happens, these variants not only associate your risk with type 2 diabetes, they also have an effect on type 1 diabetes. Very interesting, because that's telling us something about the pancreatic beta cell. In my lab, we were able to determine that those variants resulted in a loss of the protein function. So that was an important clue, because it told us if we wanted to study this protein, we needed to look at what happened when you lost that protein. So we manipulated the genes in a human islet, and we were able to show that when you lose this protein, you reduce the ability of the beta cells to make and secrete the hormone insulin. When we looked at this in more detail, trying to work out exactly what was happening, we were able to show that there were defects in the insulin secretory granules in beta cells from patients who have these variants. What we haven't published, and what we've been working on over the last year, is trying to work out whether or not we can use this information to improve how we might treat patients with type 2 diabetes. And Mahesh in my team, a clinical fellow, for his PhD project, was able to show that if you have this particular DNA change, you're resistant to an incretin, a hormone that your gut makes called GLP-1. Now, I don't expect that means much to you, but if I was to tell you that one of the most common ways we treat patients with type 2 diabetes at the moment is to use a GLP-1 receptor agonist, so working directly on this pathway, it might start to make sense as to why we were interested in following this up. Because if you're resistant to GLP-1, trying to give a patient more of that GLP-1 is unlikely to be as effective as it is in somebody who doesn't have that particular problem. And it turns out that that is the case. If you have a PAM variant, you do not respond well to a GLP-1 receptor agonist. And this means your doctor can now make the decision to put you on a different type of diabetes medication, one that's far more likely to reduce your blood glucose levels. So we're really excited about this, and also other work we're doing in this space that might help us use genetics to tailor treatments for patients with different varieties of diabetes. So I've told you a little bit about what we've been doing, what we're currently doing, and I want to finish up by telling you what my vision is now that I'm here at Stanford, and how I would like to see my research program develop under David's mentorship and within the Diabetes Research Center here at Stanford. I would like to get to a point where every single patient that David and his clinical colleagues see here has a genomic analysis, so that we can really capitalize on the huge advancements that we continue to make in our understanding of using DNA genetic variation for precision medicine. I would like Stanford to be a world-leading center for precision medicine in childhood diabetes. I would also like to lend my expertise and the discoveries that we make within our laboratory to help my colleagues in the Diabetes Research Centre fully optimize protocols for making pancreatic beta cells in a dish so that these can be transplanted into patients with type 1 diabetes. Over here, you can see some budding young scientists from a school back in Oxford. These ones at the bottom here were making beta cells, not in a dish, but in the classroom. And up here, we've got children learning about DNA and how it folds within in a pancreatic beta cell. So let me wrap up by thanking the funders, particularly the Wellcome Trust, who've supported me for the last 10 years, thanking my wonderful team, some of them back in Oxford still, some of them who've travelled here across the Atlantic to be with me at Stanford. Here we are on a Zoom call, just like today. This is how we have to meet at the moment, but we managed to make it fun and to keep it interesting. And I also want to wrap up by reminding you about my group of very close friends from my undergraduate days back in Surrey. Here's Helen, here's myself, Jenny and Sarah getting together a few years ago. Helen, I'm happy to say, has fulfilled her ambition, has a family and has 2 wonderful children. At the moment, she's healthy and doing really well, and neither of her children, she's delighted, have so far developed diabetes. So with that, I'm going to hand you back to Meg, and I look forward to chatting with you and taking your questions when we're ready. Thank you.
Well, thank you, Anna. Thank you so much for soldiering on so we could hear that wonderful update about your research. We're going to move on to the last section of our talk today. That is the Q&A section with our audience. I encourage everyone to click that Q&A button. Now is your chance to ask live questions for David and Anna. I'm going to start with David. The first question we have for you is, how did COVID-19 pandemic impact the pediatric diabetes clinic?
Yeah, well, that's, obviously top of the mind for all of us. It required us to move quickly. I think some of the work I told you about in preparing with telehealth, one of our faculty members, Priya Prahalad, is also part of the IT program and had been developing our telehealth. We moved pretty quickly then to transition visits from being in person to doing them via Zoom. That's been a progression, and I think we're getting better at it. It requires people to be able to download their devices, tools like Tidepool, others that people are able to get that data from their devices so that we can use them and then we can discuss them. I think at this point, we're still working to make it go better, but I think all of us are more used to a Zoom world, talking to each other on screens. I think the younger people are. I think a lot of our patients are comfortable with it. I think us who have gotten with it by necessity. A couple of the things that we've done in particular to change our workflow is that we've integrated interpreters into the workflow so that because we realized that we weren't reaching all of our patients who didn't speak English via this. And so now, for example, we're pretty close to the number of patients who are Spanish speaking, who are doing telehealth is very similar to our population of Spanish speaking patients in our clinic. The second thing is we're very concerned with moving to telehealth that there might be some patients who would get left behind who didn't have the ability to connect via without having Wi-Fi or other things. And what we've seen is that again, that the percent of patients who are on public insurance who do telehealth is very similar to our clinic population. So those are a couple of areas where we've tried really hard to use telehealth to bring equitable care to everyone. We still have a lot of improvement that we would like to do and make it more efficient for people. But I think this is part of diabetes care going forward. I think we'll still like to see people at least once or twice a year. But I think telehealth is going to make it easier. Even for people who live close, it can save you 15, 20 minutes in trying to find a parking spot. So I think it's here to stay. and we're working hard to make it even better for people.
Thanks, David. The follow-up question to that, knowing that a lot of patients in the clinic live over 50 miles away from a Stanford clinic, how do you think digital health will play a role after things return to normal, after the pandemic ends?
Yeah, absolutely. I think the farther away you live, the more time it saves you, and that's very, very clear. I think even people who live close can save them some time as well. I think there can be more flexibility in times for having clinic visits. So I think it's going to be something that is perhaps a silver lining from COVID. And then I think we've also done other things. As far as COVID, we've I know I did a JDRF webinar, I think back in March or April, was author on a recent diabetes care commentary on COVID and children with diabetes, with the message that it's important not to extrapolate a lot of what we hear in the media on the increased risk that COVID has for problems for people with diabetes, that's data that are in older people. And we're just not seeing in the pediatric population, we're not seeing that there's that increased risk with diabetes. Now, we want everyone to have really good control of their diabetes, but there's not that increased risk for problems from COVID that you hear about in adults. So I think that's a really important message. There were a lot of families early on, and I think still, who were very worried about that. Now, of course, wash your hands, wear a mask, and stay out of large groups and all of the public health guidance, but it shouldn't be an added risk because your child has diabetes.
Well, thank you for sharing that silver lining. We will take it. I'm going to transition to Anna. The next question is for you. What are you most excited about that you're working on currently in your research?
Ooh, always difficult to say one thing because there are always lots of things that we're working on that excite me. But having spent the whole of my research career in genomics, I have to say at the moment I am excited about the point that we have finally reached with how we can use genetics. When I look back at what I was doing as a PhD student and what I am able to do now, I realize how far we've come and that there are real opportunities to use this information to improve patient care. So hopefully with some of the examples I delivered today, you can see just how far we've come in terms of our ability to be on the cusp of really using genomics at point of care, rather than this being something that is for a small number of patients who have particularly rare genetic disorders. This becomes something that becomes mainstream and that we can use for everybody who has diabetes. And I think that's a reality now. And I particularly appreciate the fact that I have a link at Stanford to be so close to the clinical setting, to attend division meetings and to discuss how we can innovate clinical care pipelines by taking what's cutting edge in the lab at the moment and seeing how it will be implemented in a clinical way. So yeah, that really excites me, but I wouldn't say it's the only thing that really excites me.
Well, thank you. You actually answered the first part of the next question. The second piece is there a reason why someone wouldn't use precision medicine after a diagnosis? We talked about genomics testing, but this is specifically a question about precision medicine, and it may be related to your slide about drug targets.
I don't think there's a good reason for why you wouldn't. I think there are barriers to overcome because this would be a new way of doing things. And whenever there's a new way of doing things in a clinical setting, there are changes in protocol that need to be discussed. There are consensus agreements that need to be delivered. At the moment, I'm really fortunate to be part of the American Diabetes Association consensus working group for developing those guidelines for implementing precision medicine into clinical care. And I think the American Diabetes Association is showing a lot of foresight here in terms of gearing us up, because they can see that we are very close to being able to use this information, and we want to make sure that frameworks are put in place that can be rolled out within hospitals and in primary care, so that we can use this information for patient care and precision medicine.
And just to add that I'm on a different working group. They've got both of us working on this, Anna, and I agree with you. I think it's a really important initiative. I think we've historically talked about type 1 diabetes as being one type of condition, but as we learn more about the genetics, every person is unique. There are likely some differences, and we're learning more and more about that, and how can we then learn more about that and be able to tailor care more appropriately. I think looking at it another way, you think about what we're doing with an artificial pancreas system, that you're getting information about what a child's glucose is in real time. And then you have the ability with that pump and that algorithm to adjust insulin a little bit up, a little bit down, to give you what you need when you need it, and to reduce the burden of care. There are a lot of families who, historically, before these systems came out, they'd be up all night checking, doing finger pokes, giving a little bit of insulin, maybe giving a little bit of sugar to bring the glucose up. These systems now do a pretty good job of that. And that's another, I would say, form of precision medicine that's already there. It's out. There's new systems that are going to be coming. When I showed that slide of the history of the development of how we monitor glucose, how we give insulin, and now how we combine that, it's going to keep getting better and better. You can in some of my talks, I'll have a slide with kind of a history of cell phones, and these are technologic challenges. They're going to keep getting a little bit better, a little bit better, a little bit better, and we hope a little bit smaller and a little bit user-friendly. But they're already a whole lot better now than even 5-10 years ago. And I think these are engineering challenges that I think are, people in this area for sure are very savvy about technology. It's not perfect. It's not a cure, but my goodness, it's certainly making life better for so many people.
Well, I think this next question is for both of you. We already know that we're a part of JDRF's Northern California Center of Excellence grant, and this is a Bay Area collaborative for diabetes research. Anna, you also mentioned the islet cell replacement program that's part of the Bay Area. Our next question is of one of dreams towards the cure, I think. The first step is, how far are we from islet cell transplants in the road to the cure for type 1 diabetes?
Yes, it's a difficult question to put a time frame on. I'm not avoiding, that I perhaps 10 years ago would have thought this was a complete dream, and it was such a long way off. But the way that the field has moved in recent years is making me reevaluate how possible this is. I think if you look, for example, at what CRISPR has done for the field, our ability to use these molecular scissors to cut out genes, to make different versions of them, it's really changing the way that people can adapt protocols. We've got some fantastic scientists here in the Bay Area and in Stanford in particular, who are tackling some of the really large challenges, I think, in terms of stem cell replacement therapy, which is the immune system. How do you get a new replacement beta cell into somebody who has an immune system that's going to do everything it can to get rid of that beta cell? And there are some exciting ways that this can be approached now using CRISPR. So who would have thought 4 or 5 years ago that we would be in this place now? Who'd have thought we could use molecular scissors? So I feel very encouraged about the speed at which the field is moving forward. And I think what needs to happen is to make sure that we have integration of interdisciplinary teams. So when there are advances in technologies, they are adjacent to people who are asking the big and difficult questions for medical health, so that they can be rapidly adopted. And going back to my slide about why Stanford, that's something that I very much see happening here at Stanford, is the innovation with technology next to the clinical need and biological expertise. Put those three things together and it's really powerful for shifting a field rapidly.
David, do you want to
I
answer?
absolutely agree with you, Anna. I think we always want to be cautious when we're setting a timeline to bringing discoveries to the clinic, because we don't want to let people down. I think there's probably people listening right now who had someone give them in 5 years, and it's hard to predict, and some of it's out of our hands as far as when something's going to get approved or what the developments are going to be. I think we can say that it's going to be better in 5 years. I think, as Anna said, there's a great team here in the Bay Area, elsewhere, working really hard. It is that combination of we have to get the beta cells, and there's great work being done to get stem cells to beta cells. But then it's the immune system as well. So if we put the beta cells back in, we have to do something to turn the immune system off and turn it off selectively, so that it leaves the new beta cells alone, but it still fights off cancer cells, flu cells, strep throat, other things. So how do you come up with that medication? And then I think a lot of the work that's being done with the diabetes technology, it's not a cure, but it is reducing the burden, and it is getting the glucose control better and better. So I think until, and we will keep working to try to make beta cell replacement a reality, but it's probably going to start in older people and then move into younger people as it's shown to be safe and shown to be effective and more effective. And then there's going to be some people who want one treatment versus the other treatment. And I think that's a goal that we get to the time where people have different options to take care of their diabetes the way they want to.
Well, thanks, David. We're on our last question for the Q&A. People submitted some great questions. What role can philanthropy play? And we're running out of time, if it can be brief.
Anna, do you want to take that first?
I'm happy for you to. I'm just answering
Okay.
the question in the chat.
Okay,
oh, perfect.
Good. So I think, I think, Meg, much like we talked about the NIH Diabetes Research Center grant, that's really brought together the research community on the Stanford campus. We've been able to attract new people, brilliant young scientists to do work in diabetes. We've also been able to attract more senior people who are brilliant scientists, have brilliant research methods, but haven't particularly thought about doing research in diabetes before. And so with some of the pilot feasibility grants, those have helped get people in the area. So philanthropy can accelerate the research that we're doing. Philanthropy can accelerate the translation of basic discoveries into the clinical setting. And it's really, really important to be able to have that type of support to make us go faster, to get better care in the clinic, to be able to prevent diabetes, and ultimately we want to be able to cure diabetes as well.
Well, thanks, David and Anna. We are coming upon 5 p.m. As a final reminder to the audience, if we didn't get to your question in the Q&A section, we'll follow up after the event. Thank you, everyone, for joining the webinar today. We are so grateful that we could gather, even if virtually. I want to thank all of our speakers, Dr. David Maas, Dr. Anna Gloyn, and Melissa Anderson. We appreciate you taking the time to share these important updates about the mission to both care for and cure diabetes. As we continue to raise funds for this important work, we all hope that we can count on you today. Our diabetes program's growth, as David has shared, would not be possible today without the generous support of donors like you. And we believe that these partnerships truly push boundaries in the field of diabetes. So please look out for an email in your inbox after this event. There will be a Zoom recording. There'll also be contact information for Lucille Packard Foundation. If you're interested in learning more about how you can support the field of diabetes at Stanford, we would love the opportunity to speak with you. If you have any questions, please reach out to me and I can connect you with someone at the foundation. Thank you again, everyone, and have a wonderful evening.